Thank you very much. Thanks a lot. I have a micro. Well, it's a pleasure to be here. Thank you. I will use the opportunity to introduce you to a Swiss-wide incentive on molecular surveillance called the Swiss Pathogen Surveillance Platform. And this platform, as you can see, is an incentive supported by all Swiss universities and Swiss university hospitals. But before I go into this, I'm also representing the Institute of Medical Microbiology. So I would like to briefly introduce the Institute to you. And I really think this is the best of both worlds. On one hand, on the left side, you can see our routine diagnostic teams, where we have different specialists in various areas like general bacteriology to serology. And on the other hand, we also have a profound research expertise, and you can see in the middle part, that's my research group. And if you want to collaborate later on with me or with other groups in the Institute, please reach out. Also, Marco Seger is part, obviously, of the Institute, but we have a lot of other fantastic colleagues to work with. So let me dig into molecular surveillance. So you're all aware of that multi-drug resistant and virulent bacteria and also viruses and fungi are emerging, and this strongly impacts humans and the veterinary field in terms of morbidity, mortality, and healthcare associated costs. Now, how to best interrupt this is obviously you need to have a good idea where transmission is happening, but the problem is that this is very complex. And one technology which really changed here the field is whole genome sequencing, because it provides highest resolution data, and you can really link this with a lot of meta information to get also spatiotemporal resolution. There are different compartments: human, animal, food, and environment, as we've heard before. And I just brought you here a couple of examples. On the very left, you can see a surveillance program in Basel on Campylobacter, where we want to butcher stores and bought chicken meat and linked the isolates from chicken meat with the clinical isolates we had. In the middle, you can see a Legionella pneumophila outbreak that was from air-conditioned cooling towers in the city, causing an outbreak, a small outbreak in the city. And on the very right side, you can see a refugee-associated outbreak of Corynebacterium diphtheriae, where we had 360 cases in 10 European countries, and this was managed together with the ECDC. And very dangerously, we also faced erythromycin resistance. Okay, to generate such a surveillance platform, what do we need? We actually need a platform where we can exchange whole genome sequencing data with additional information. And this data has to be interlinked, first of all, with other platforms, analyses. You will hear later on more about them, but then also to other surveillance platforms in Europe. And metadata provides you the context. So this metadata has to be in a very structured way, very controlled vocabularies are used, and this is what is required. So how do you get this? And here, Switzerland has invested quite a lot into the data infrastructure. This is called the Swiss Personalized Health Network, SPHN. And what is SPHN? This is a network under the guidance of the Swiss Academy of Medical Sciences, and together with the Swiss Institute of Bioinformatics, it contributes to the development, implementation, and validation of coordinated data infrastructures which can be used for healthcare. And SPHN obviously builds and supports then existing data sources and infrastructures across the country. And this was used for various aspects of healthcare, oncology, but also infectious diseases. And I want to bring you here two examples. The first one, where I will spend most of the time on, is the Swiss Pathogen Surveillance Platform. But in one slide, I also want to show you the national data stream called IICU, where also Annelies Tinkernagel, for example, is involved. So, SPSP, how does this work? So first of all, you have here the database, which is centralized at the Swiss Institute of Bioinformatics. Individual labs can upload genome sequencing in a very secured way with a two-factor authentication. The data is then quality controlled. An analysis step happens across a bioinformatic pipeline where you have assembly of the data, typing, and tree generation. The data is visualized. I'm also going to show you a few things there. And then you can manage the data by, for example, sharing it to international data repositories, and this is automatically done, so we share a lot of our sequence data, but you can also generate custom reports to federal offices and all sorts of experts. Initially, it was funded in the NRP 72 program as an idea to monitor methicillin-resistant Staph aureus, but then with SARS-CoV-2, with the pandemic, we actually switched gears, and it was the main data sharing hub for SARS-CoV-2 data. What you also need is a legal, ethical, and governance framework, and that took us a while to set up. So we have a consortium agreement between all these partners. We have a data transfer and use agreement, and we have an ethical approval, so the data can also be reused for research purposes, and I think that's really an achievement. And many European colleagues actually ask us, how do you manage that? And we've shared this framework, the idea and the templates many, many times since then. So it works on a regular basis. We have once a month an executive board meeting, we have regular scientific board meetings, and there's also an advisory board where, for example, also the Federal Office of Public Health is in there. So these are just the different centers. You can see across Switzerland, everybody is participating here. And also we reach now more and more out to European partners. So for example, there's a current project on VRE surveillance in Germany and Austria. And then we also participate in another incentive with national reference centers. Just one word on the pandemic, because this is really our showcase. You see here the positive PCRs in Switzerland over time. So you have here a time axis. Here you see the PCR results with the different waves we faced. Then here you see the amount of sequencing, and the stat ends here in 2022, but since overall we have sequenced more than 170,000 genomes, and all these genomes went via this platform. And so we've sequenced 7% of all the PCR positive cases, which is massive for the size of the country. We have a very, very high sequencing density. Now, maybe we've sequenced too much, and that's actually also a question which we ask ourselves, which I will answer in a second. Here you can see the public dashboard, which you can access. If you go to the website, you can explore the data yourself. Currently, we have the virus part with SARS is active, but more viruses will come. For example, influenza will also go in there, and a couple of bacteria. So how much sequencing is enough? And this is actually nice. Now we can access this data. And what we've done with all these sequence data, we did random downsampling 100 times. We picked different sample sizes. So you can see here on top, if everything was sequenced of the Alpha, Delta, and Omicron variant, we wanted to know if we sequence less, do we have a time delay in finding a new variant? And here you can see on the x-axis, that's the days delay of picking up then this variant. So with a third of the sequencing, we would still have had no delay. So that's really an important message. Obviously, we really sequence too much, and that's important to actually know, and you can only answer this question if you sequence too much, because else you cannot downsample. And I think that's one of the projects we have here. So what's coming next? We develop further the front-end part with the dashboards, as I mentioned to you, the queries. We will also expand it to a series of bacteria. And just a couple of weeks back, we got financing confirmed from the Federal Office of Public Health. So we will focus on a couple of foodborne pathogens. We also have funding for bacteremia as part of this sepsis network, which I will also show to you. And then we have a few research-driven projects. What is also very interesting, if you're interested, now addressing that to the Indian colleagues here, to use this platform and download the programmed platform, you can actually do that. And we would be very happy to help you implement that here. So I think it's a really nice platform. We also have bacterial data on it, and here you can see the number of species. So a lot of bacterial species have been sequenced. Now, obviously, there are also very rare species in it. Here you can see the numbers of the actual sequenced species. So we have roughly 3,000 Staphylococcus aureus in there, roughly 1,000 Enterococcus faecium in there, and Klebsiella pneumoniae, maybe 800. And we have also very rare ones, like Mycobacterium basiliense. That's my most favorite bacteria you've probably never heard of. I discovered it and I named it to honor my hometown. So it's Mycobacterium basiliense, 5 cases, and this is also in the database. So if you find it, you could actually easily match this up. So that's the fun part of being a medical microbiologist. Sometimes you can name a bacteria. The bioinformatic pipeline behind that, that's a next flow-based pipeline. It's very modular. You have different steps of quality control, and then you can do some typing part, some taxonomic part. Also, AMR identification is in there. And then we have different ways how you can report. Just my second last slide here. That's now, I switch gears. That's the sepsis network I'm going to show you. That's also a Swiss-wide network. Has nothing to do directly with surveillance, but this is also a data-driven incentive where we have different clinical data warehouses in each university hospital, and intensive care unit data is shared to the ETH domain, to Leonhard Met. That's a highly secured environment for data analysis. And we have collected so far data of 40,000 patients on the intensive care units. Just to give you a flavor, we have there close to 100 million temperature measurements of these patients on the intensive care unit, and all sorts of lab data, physiological parameters. And currently we use artificial intelligence to predict sepsis appearance. That's a fantastic network, and here partners again with Annelies Zinkernagel and Reto Schüppach from the intensive care unit in Zurich, but obviously a lot of colleagues in Switzerland. So in conclusion, what I've shown you is that SPSP really allows data sharing of molecular and classical epidemiological data. We follow the FAIR principles, so we make our data interoperable. We follow ontologies to be able to share the data. Next steps is to focus on a series of other pathogens. We want to connect with different incentives of EFSA and ECDC, and we also now interlink this with the ICU project. And if you're interested, please reach out to me. So here you see the colleagues involved in SPSP, so a lot of university colleagues, but also federal colleagues and many other labs who share data over this platform. And with this, I'm finished, and I thank you very much for the attention.
Thanks a lot. You've been perfectly in time. This was a very nice big data promoting talk, and I think, is there any question? I think we have one question. Is that okay? Because in the interest of time, because there's lunch coming up, and so I give.
Thanks, Adrian.
To you. Is that okay?
I was just wondering whether the platform would work if hospitals are not using. I'm Shraddha. I work at Ashoka University High, and I was wondering if the platform would work if hospitals are not using terminology SNOMED or anything, because I try to work with them a bit, and turns out most of the hospitals are still not using any terminology.
It would work. The thing is, what we have to collect the metadata is it's really simple. It's an Excel sheet where you type in the data, and there you have a forced vocabulary. So you cannot just type in free text.
Okay.
So it generates, the same wording, and that's one of the key advantages, I would say.
Thanks.
But we can further chat about how you would implement that in your system. Thank you very much.